see something now.
Yes, Professor Jia, good morning.
Good morning to you.
Okay. So everybody will be assembled very soon.
এখানে যা পাবো অ্যাডমিট করে দেবো তো
Hello, Professor Jia, I am Satrajit. Hello, Professor. How are you?
Hello.
Hello, I am Satrajit. Satrajit. Hello, I am Satrajit. Hello, I am Satrajit.
Oh, hello. Yes.
Yeah. How are you?
Fine, thank you.
Okay. So I remember that once in Pondicherry there was a conference we met in a and then
That was an SDLC meeting.
SDMC meeting.
Mhm.
Okay.
Long ago.
Nice to meet you again.
Good to see you.
Okay. Good morning.
And that's a good evening.
Oh, that's a good evening. Sorry.
It's my evening. It's your morning. Yes.
Yeah, my morning. Yeah.
So I think all is working well. Yeah, we're connected.
জয়েন করে আমরা করে জয়েন করে দিচ্ছি জয়েন করে দিচ্ছি ওটা তো আমরা এখান থেকে দেখে নিচ্ছি আপনাদের
ঢুকবে টাইম মতো, ও একটা মিটিং মেয়ের মধ্যে রয়েছে, কিন্তু ঢুকবে। আমরা কয়েকজনকে ফরওয়ার্ড করতে পারতাম। আমি সত্যমুখে করছি।
Good to see you.
Good to see you too. How are you doing? It's been a while.
Fine, thank you. Fine.
I missed you.
I'm at home.
Oh yeah, I can see. I missed you when I went there back in fall. That was before everything.
Yeah.
So you're looking good. It doesn't
Thank
look
you.
like you're aging at all.
Oh, I am. Looks are deceptive that way. Okay.
Especially in a Zoom meeting.
I really have some new data for you in this.
Oh,
this
really?
talk.
Oh.
I'm excited.
Yeah, look at your lecture titles are always exciting. So let me see what a drop can do. So are you still in mud or you have moved?
No, well, I'm still in mud. They're planning to tear it down, and we will move to another.
Yeah, I know. I
building.
heard about it last time. I mean, I was sort of not happy about how they got rid of the lawn in front of it, and the old gazebos are being moved. Yeah, I did like the old chemistry building being renovated for library. That's really great. That was awesome. But they
Yeah.
got rid of the Swen building, and I used to sit in OC, right? So now OC is gone.
Right.
ফরওয়ার্ড করো, যে তুমি কথা বলো।
So, right, I'm going to hear your lecture in a minute. Now I'm going to turn off my video. Just wanted to say hi. So,
Oh, appreciate, no.
এই ওয়েটিং রুমে ওয়েট করছি, একটু অ্যাডমিট করে দাও।
I really wish I were with you.
Yeah,
It's-
you are with
Okay.
us. You are with us. No problem.
Good evening, Dick. How are you?
Fine, thank you. Good to see you.
Yes, you visited, I am from Indian Institute of Science, and you visited several times. We had several meetings together, Pushpendu, myself,
Yes.
Uday, and yourself. We had a lot of chats. I hope you remember that. And I am here for last over 5 years, as the head of the institution.
Right.
So actually, I was thinking of having you here physically, but
I wish it could be.
yes, that would have been fantastic.
The coronavirus situation is not good in California. We
Yes.
think we're getting better now, but it's been very bad.
Yeah, I know that because my daughter stays in Los Altos, which is the next
Okay.
town of Palo Alto, where you live,
Right.
and I keep update almost every hour.
So I live on the Stanford campus.
Yes, that's
Actually,
in Palo
my
Alto.
home.
I know. Yes, but I hope situation is getting better.
It will. With the vaccination, it will.
Okay.
But there are still people who insist that somehow public health infringes on their private freedom, and so they don't obey rules.
Yeah, I just read in today's CNN, very strange thing. They are actually advocating some groups, particularly the churches that don't take the vaccine because they are originating from fetal cells. And, how do you control that? So I think this is around 9:30 your time in the evening.
Yes, yes, it is.
Okay, we'll start in a minute or two. Suppose.
I see Nanamani's with me, though can't see his face, but I see he's there.
Hi, Dick.
Hi,
Yeah, I'm here. How are
great.
you?
I'm glad you could join.
Yes, I will be happy to join your lecture. I hope everything going well with you.
I can't see everybody who's here because I don't have the, yeah.
Yeah, maybe some of them, like turned off their video.
There are 27 people, so there's at the
Hi, Dick.
moment.
Srihari here.
Oh,
Hello.
good.
Good evening, Professor Zier. I'm delighted to have you here online on behalf of our institution, Indian Association for the Cultivation of Science, to deliver prestigious Jagadish Chandra Bose Memorial Lecture instituted in the honor of the legendary scientist Sir Jagadish Chandra Bose, a biologist, physicist, botanist, and early writer of science fiction. He pioneered the investigation of radio and microwave optics, made outstanding contributions to plant science, and laid the foundations of experimental science in the Indian subcontinent. The Indian Association for the Cultivation of Science, acronymed as IACS, was established in 1876 by Dr. Mahendralal Sarkar. IACS was the first national science association of India. Basic science departments such as chemistry, physics, mathematics, physiology, botany, geology, etc. were established, and notable Indian scientists participated in the association. Regular lectures and demonstrations were arranged for the public to popularize science. Dr. Sarkar supported women's education in 19th century India, when higher education among women was indeed rare. IACS is also the oldest research laboratory of Asia, founded 145 years ago with the financial support of the philanthropists and nationalist leaders. It is where Sir C.V. Raman joined, started his laboratory, and discovered the well-known Raman effect, which won him the only Nobel Prize in Science from our country. Satyendranath Bose, famous for his pioneering work on Bose-Einstein condensate, was National Professor in Physics in the Theoretical Physics Department of this institution. And the modern IACS started its present campus with Professor Meghnad Shah, who is well known for his celebrated Shah ionization theory, which is applicable in stellar bodies, as the first official director. Today, IACS is one of the major research centers of India, working in several frontier areas of physics, chemistry, biology, mathematics, computer science, material science, and interdisciplinary sciences. Department of Education in the year 2018 has conferred a deemed to be university status to IACS on the recommendation made by the University Grants Commission. IACS organizes a number of endowment lectures named after the luminaries of Indian science associated with this institution and renowned internationally. Previous speakers in such endowment series included world-famous professors Roald Hoffmann, Ahmed Zuel, Gerald Toft, Wolfgang Ketterle, Yuanti Lee, H.E. Negishi, Ben Feringa, among others. I deeply appreciate that you have most kindly accepted the invitation and have agreed to deliver the J.C. Bose Memorial Lecture for the faculty, staff, students of the institution today, also as a part of the celebration of the Golden Jubilee of the Department of Science and Technology, the Administrative Ministry of IACS. The lecture will be attended by numerous schools, colleges, and universities as well. Now, I introduce today's J.C. Bose Endowment Lecturer, Richard N. Zaire. Dick Zaire is the Marguerite Blake Wilbur Professor in Natural Science at Stanford University. He was born on November 19, 1939 in Ohio and studied from Harvard University, where he received his BA degree in chemistry and physics in 1961 and completed his PhD in chemical physics in 1964 under the tutelage of Dudley Horsback. In 1965, he became an assistant professor at the Massachusetts Institute of Technology, but moved to University of Colorado the next year. And after few years there, he came back to the East Coast again and accepted a full professor position in the Department of Chemistry at Columbia University, becoming the Higgins Professor of Natural Science in 1975. In 1977, he decided to move to West Coast and he accepted a position in Stanford University. He served as chair of the Department of Chemistry at Stanford in 2005. And in 2006, he was named a Howard Hughes Medical Institute professor. Dick is actually continuing in Stanford University since then. He's renowned worldwide for his outstanding research in the area of laser chemistry, resulting in a greater understanding of chemical reactions at molecular level. By experimental and theoretical studies, he has made seminal contributions to our knowledge of molecular collision processes and contributed immensely to solving a variety of problems in chemical analysis. His development of laser-induced fluorescence as a method for studying reaction dynamics has been widely adopted in other laboratories the world over. Dick also worked with NASA and other astrobiological problems. He's one of the co-authors of a paper of science that appeared in Science in 1996, raising the possibility that a meteorite from Mars contained traces of Martian life. He's also involved in the development of desorption electrospray ionization techniques, which are being used for mass spectrometric imaging of lipids, metabolites, proteins in tissue samples, including prostate cancer. In his own words, quote, "I am right now very excited about mass spectrometry, still about lasers, all types of analytical techniques, but to me, they're tools. They're not ends in themselves. With new tools and measurement techniques, you can make advances in all types of fundamental problems," unquote. Professor Zaire has given numerous named lectures all across the globe, has authored and co-authored close to 1,000 publications and more than 50 patents, and has published several books. Consistent with such an outstanding profile, Professor Zaire has received numerous honors and awards. If I actually go on, this will be a very long list. I will just mention some, the notable ones. Michael Poliani Medal of the Royal Society of Chemistry in '79, National Medal of Science 1983, APS Irving Langmuir Prize 85, National Academy of Sciences Award in Chemical Sciences 91, ACS Peter Debye Award in Physical Chemistry 91, the Harvey Prize 1993, Linus Pauling Award 1993, the Centennial Medal, Graduate School of Arts and Sciences, Harvard University, the Wells Award in Chemistry 1999, ACS Nobel Laureate Signature Award for Graduate Education 2000, Royal Society of Chemistry Faraday Lecture 2001, Chandler Medal, Department of Chemistry, Columbia University 2005, the Wolf Prize in Chemistry, Israel 2005, the Dudley R. Hirschbach Award for Excellence in Research in the Field of Collision Dynamics, F.A. Cotton Medal for Excellence in Chemical Research in 2009, King Faisal International Prize in Science 2011, Honorary Membership of the Japan Society for Analytical Chemistry, Einstein Professorship of the Chinese Academy of Sciences. And Dick has been one of the most outstanding teacher as well and received numerous teaching awards, both Stanford and nationally and internationally. I would only mention that he's not only received several times the Dean's Award and Bing Fellowship Award at Stanford for his undergraduate commitment to teaching towards undergraduate education. He also received James Flack Norris Award for Outstanding Achievement in Teaching in Chemistry, ACS George C. Pimentel Award in Chemical Education, and Presidential Award for the Excellence in Science, Mathematics, Engineering, and Mentoring. He also holds several honorary degrees from Arizona, from Northwestern, from ETH Zurich, from Uppsala, from Columbia University, from University Paul Sabatier, Hunan University, Chalmers Institute of Technology, and he is the member of National Academy of Science, American Academy of Arts and Sciences, American Philosophical Society, the World Jewish Academy of Sciences, Fellow of the American Physical Society, American Association for the Advancement of Science, the Association for Women in Science, the American Chemical Society, the Honorary Fellow of California Academy of Sciences, Royal Society of Chemistry, Chemical Research Society of India, the Indian Academy of Sciences, Fellow of the World Academy of Sciences, and Swedish Royal Academy of Engineering Sciences. It is my great pleasure to invite Professor Zehr to deliver his lecture.
Thank you so much, and for this very fulsome description of things I've been doing. I'm going to share my screen now and see if I can make this work. Okay, move that there and go here. And now I want to go from slideshow. I'll push this and I'll go from the beginning. Here we go. So indeed, I'm delighted to be selected by you to be the J.C. Bose Memorial Lecturer this year, and appreciate this very much. I have actually been to IACS back in February 7th, 2014. And here's some pictures I took then. I was able to give at that time the C.V. Raman lecture and had a great time, stayed in your guest house with my daughter, Bonnie, who was with me at the time, who also, like me, loves India. I'm going to talk to you about microdroplets, small droplets of water. To begin with, let me talk about water itself. It's so common, but actually so mysterious. You think you understand a lot about water. It's all over the place. Find it everywhere, but we're still learning things about water. Let's consider a 1 liter beaker of water. That's shown here, okay? And you might ask the question, what does it contain? Well, you learn very quickly in beginning chemistry that it's actually, if it's 1 liter, it contains 55 moles of H2O. And mole being Avogadro's number, something like 6 times 10 to the 23, huge number of water molecules in this. And you might ask, well, does it contain anything else? And if you think about it, well, yes, actually, it does contain a small amount of H plus, okay, the hydronium ion, and OH minus, the hydroxide ion. And the H plus amount is like equals the OH minus amount, and at room temperature and 1 atmosphere pressure, it's about 10 to the minus 7 moles. Very different, very small amount. Okay. And indeed, all beginning chemistry students learn that the pH, which is the negative logarithm to the base 10 of the hydrogen ion concentration of normal water, is just 7. Everybody says that. That's neutral water. Okay. But is that the end of water? We know water is very unusual. We know, for example, that it requires a much higher temperature to boil. And we really attribute all this to hydrogen bonding in water, and that things make a network and stick together. The very fact that ice floats on water, that's the solid form of frozen water, okay, tells us that ice is actually less dense, less compact than the water with the hydrogen bonding that's snuggled together and get quite close. Suppose, though, if we ask the question of someone who's beginning in chemistry, well, you tell me that the pH of water is 7. Have you ever thought about what is the pH of a water droplet in air? And the person looks at you puzzled because you normally would expect the answer would be 7, but you're asking this question, so there must be more to it. And if you think about it, there is. First of all, at the surface, you no longer have 3-dimensional hydrogen bonding. And this is going to really lead to all types of interesting effects. You now have the possibility of water moving around on the surface in different ways and being much more ionized. And no longer is it necessary for the pH and the POH to add up to 14. They don't have to be each the product 10 to the minus 7 times 10 to the minus 14, 7 being 10 to the minus 14 for the kW of water. And let's go further with this and look at it. Here's a model. Because we have ions at the surface and water itself is not symmetrical, okay, it's a polar molecule itself, we expect that there's going to be some end that prefers to stick into the hydrophobic medium, whether it's oil or it's air, that doesn't like water, hydrophilic water. And so this is a model of a water microdroplet in air or oil. And the result is to set up a double layer of charge. And people have found experimentally that pure water generally behaves as if it's negatively charged on the outside. There's various arguments about why that's so. Is it because the OH minus prefers to go to the outside compared to the H plus? Or is it because of impurities such as dissolved carbon dioxide that make the bicarbonate HCO3 minus ion? Or is it because just the nature of water itself organizes itself with the ON sticking into the hydrophobic medium and leads to this charge distribution? That's still being actively discussed. But it's clear that it's now very different. So why does water microdroplets differ so markedly from bulk water? And they really do. They're not the same. They as I show you in a moment, and they're remarkably different. There is a huge electric field at the water microdroplet periphery. Go back. You're making a separation of charge, and I'll show you there's a very large electric field on the order of about 10 to the 7th volts per centimeter. You can't generate such a field generally in the lab without having some type of lightning strike if you try to do it with condenser plates. But water droplets have that. There's, as I say, a reduction in 3-dimensional solvation and hydrogen bonding at and near the interface. And there's going to be an increase in the autoionization, yielding both more H plus and more OH minus concentrations. And this will cause the acceleration of the rates of both acid catalyzed and base catalyzed reactions. And you don't think of that normally, but this is what's going to happen. It's quite possible that the kW of water at its surface is really changed by many orders of magnitude. And we have some evidence of that. And the concentration of reagents at the interface and partial desolvation all contribute to make reactions go at the interface of air water or oil water, which won't go in bulk water. So we can start to expect there'll be some differences, and I'm going to show you them, some of them. One of the prettiest examples I know is work done recently by Shivdas Banerjee at ISER Tirupati, in which he was able to show with his team the capture of the elusive carbocations. This is one of the carbocations shown to the right here. And normally, this is what George Olah made famous with magic acid, a superacid. And aqueous microdroplets act like magic acid. Amazing. And notice the publication of this was February of this year. So we're talking about stuff that's very recent. And I don't imagine everyone knows this yet, but it's really so. And it's quite impressive. And we'll have other examples. Here's an example I want to share with you. This is something that we did in a satellite laboratory I had at Fudan University in Shanghai. And this is the tryptic digestion of proteins. So in bottom-up proteomics, enzymatic digestion of proteins is an essential and critical approach to breaking down proteins into smaller polypeptides prior to protein structure elucidation by mass spectrometry. That's been a standard method of how to sequence proteins. Okay. In a typical procedure, the protein solution is mixed with a proper amount of some enzyme, often trypsin, but you can use others, and incubated overnight at 37 degrees centigrade. They pick 37 degrees centigrade because that's where trypsin has most of its activity. If you get too much hotter, the trypsin unfortunately decomposes, and colder, it doesn't move as much. So it's just right at 37 degrees centigrade. And as I say, you need hours for this process. It's easily done. And to facilitate digestion, protein denaturation is usually performed before the digestion to destroy the compact globular structure of the protein, explores more proteolytic cleavage sites because trypsin only cuts certain amino acids. And let's look at an alternative approach that I'm going to describe to you. What we're going to do is simply take a liquid sample that has the dissolved protein of interest along with trypsin in water, and we're going to shoot it out. We will or will not apply an electric field to the charged particles. You don't need to actually apply electric field, but in some cases it's better when we do. And I'm telling you the proteins will form peptides on the way to the mass spectrometer. So here we are. We're going to use aqueous microdroplets containing protein and trypsin. This is going to be done at room temperature. It makes it really easy. And it's going to be no sample pretreatment. It'll all take care of itself. And I'll show you that you can, in many cases, get complete digestion in less than 1 millisecond. That's the travel time from here to here to make this happen. So this is an amazing acceleration of a chemical reaction, I think. And it's made possible by microdroplets, by water microdroplets. And the person who's been leading this is Dr. Xiaoqin Zhang, who is now taking on her own faculty position at Shanghai University. And I want to show you the nature. This is a commercial ESI sprayer, and this is the size of the droplets. And you'll notice if you look carefully, they're peaking, I don't know, 60 microns. But our home-built sprayer peaks more, I don't know, about 9 microns. It's much smaller. This is going to be important because it's going to turn out that small microdroplets are much more powerful than large microdroplets, which is one of the reasons why a lot of this has been missed by people. They haven't seen the advantages of small microdroplets. Okay, let's take a simple polypeptide, ACTH, adrenocorticotropic hormone. It's produced from the pituitary gland in the brain, and it functions to regulate levels of steroid hormone cortisol released from the adrenal gland. Here's the sequence, and I'm showing you with these straight lines. Oh, let's come back. Show you here. This is where it's going to be cut. The vertical lines indicate the expected cleavage sites for trypsin digestion. Okay, and here is the tryptic digest of human ATCH in water microdroplets. And I'm showing you the mass spectra, okay, and the A is undigested, no trypsin, and you'll see you have these different peaks. And you have more than one charge. I'm giving you the number of charges, 3 plus, 4 plus, 5 plus, et cetera, in the peaks. Because we talk about M over Z in a mass spectrometer. The mass to Z is Z, I think to you, is the charge. And now look what happens when you add this in droplets. This is B3E digested with 5 micrograms per milliliter of trypsin at different travel distances. And I'm showing you what happens at 2 millimeters, 10 millimeters, 20 millimeters, and so forth. And we see we have basically removed all it's completely fragmented, okay? And I'm giving you the facts below as to why it's happening in 0.6 microseconds is the travel time in this case. Okay, that was ATCH, and it's impressive. But what about something else? Okay, this is now digestion with a commercial ESI. See, it doesn't work. Droplets are small. This is the digestion in bulk, 3 hours. Again, you don't get much. Doing so much better with microdroplets. Let's take a case that's really hard. This is notorious myoglobin for being indigestible by trypsin. It's the primary oxygen-carrying pigment of muscle tissues. You know, it was the first thing to have its X-ray crystallography done on it, for which I guess a Nobel Prize was given. Myoglobin contains 154 amino acids and consists of 8 alpha helices connected by loops. Okay, and now look at it. I'm showing you right here bulk phase, and you'll see the ones in green are undigested, and these in red are the fragments. And we find that the coverage is about 60%. And this is after 14 hours at 37 degrees centigrade. But look what happens if we do something else. Look what happens if we do room temperature and spray it. We completely digest all the myoglobin, and indeed you get all these peaks. And so trypsin cleaves peptide chains mainly at the carboxyl side of the amino acids, lysine or arginine. And the red asterisks denote the peptide fragments. The green numbers there mark undigested myoglobin peaks. And the coverage is 100%. And well, you say, is this a special case? Can you do more? So why is digestion, in fact, why is this happening? Why is digestion of proteins in water microdroplets so dramatically accelerated? And it turns out that trypsin works best in a basic medium. It likes to have something which is base. But proteins unfold best in an acidic medium. That's acid denaturers, they open up best. And as I was mentioning to you, water microdroplets at its surface are both acidic and basic at the same time. And so the partial desolvation and high concentration of charged species, all at the interface, lead to this. Here's another one. This is something called Herceptin, and it's a monoclonal antibody used to treat breast cancer. And it's a big thing. And I don't think I want to bore you, but rather with the details. Here it is being separated. I just want you to see very quickly that we're able to break it apart again and do great sequence coverage. And Professor Hao Chen has joined me in this work, who's now at the New Jersey Institute of Technology. Here's my graduate student, Christian Forrest Chamberlain. And the question is, well, how big is the electric field? What's it like? And so let's now pause for a moment and get quantitative, okay? Let's consider a surface density of 1 charge per 100 square nanometers on a 10 micron diameter droplet. That's a reasonable estimate or guess as to what's going on. That's not much. So what's the surface area? It's 4 pi r squared. And so putting in what is r and so forth, we get that it is basically pi times 10 to the 8th nanometers squared is the surface area. And now at 1 charge per 100 square nanometers, then we have 3 pi times 10 to the 6 charges on the surface. So far, good. And at this means they have to have opposite charges in the volume of the droplet. They have to be neutral. Droplets start out neutral, stays neutral. The charges just separate. And this corresponds to 5 times 10 to the minus 18 moles and a concentration of about 10 micromolar, okay, of what we're talking about. And so let's go further. Let's get quantitative about this. So we have here the charges times the coulombs per charge. And we find out that this corresponds to like 5 times 10 to the minus 13 coulombs. And the electric field at a radius of 5 microns, remember the diameter was 10 microns, from a sphere of charge containing these many coulombs is given by this formula that you learn in undergraduate electricity and magnetism, Q over 4 pi epsilon naught, and then you have to put in the actual dielectric constant of water, which we'll guess is around 80, okay, 78.4 r squared. And when we work all this out, you'll see that it turns out to be 2.3 times 10 to the 6 volts per meter. This is getting big, okay? It's a huge electric field strength. And you can now actually make a steady state continuum treatment of this process in which you have no flux going on. And that's a balance, the action of the electric field against diffusion. That's what I'm showing you here. And this just gives you a feeling for what's happening. And you can work this out, get the electric field, and you can assume Stokes-Einstein relations for this, get the electric field. And when you put in Gauss's law and calculate all this, and then write a computer program, you assume spherical droplet of radius r and switch to polar coordinates to get the formula like this, you can actually plot out what are the concentration of species. And I'm showing you a bunch of curves. And I'm showing you that at 10 to the minus 10 molar, nothing much happens. This is the average unpaired ions. After all, so many ions stick to the surface and the rest are unpaired, okay? But as you move to like 10 to the minus 5, they really pile up at the distance here. In this case, this is for a 20 micron diameter water droplet to 25 degrees centigrade. And we see that we really are going to be making very large differences. There are going to be gradients in our droplet, spatial gradients of concentration. And this is the radial dependency of the electric field that's been worked out. And so micron-sized water droplets might now not surprise you so much, induce spontaneous redox reactions, okay? This is going to be, again, something that I don't think you'll initially expect at all. We didn't, and it's been very hard to get this type of work published because initially no one believes you when you tell them this, but I'm going to show it to you. Here is an example. And notice all we're doing is we're spraying through a silicon capillary. There is no electric field being applied. Here's a reaction distance, and here's either a mass spectrometer or something else to collect what we spray. I hope I'm making clear. Look at what happens. Here is pyruvic acid or pyruvate, and here is lactate. We all know lactic acid. And the conversion of pyruvate to lactate in the body is enzymatically catalyzed by something called lactase dehydrogenase. In this reaction, pyruvate gains 2 electrons, becomes reduced, and is converted to lactate. And this is catalyzed with this NADH, loses 2 electrons to form NAD+. Lactate dehydrogenase converts pyruvate, the final product of glycolysis, to lactate when oxygen is absent. And that's why when you do a lot of muscle exercise, you get lactic acid buildup and your muscles hurt. Okay, fine. That's how nature has been doing this. But let's now do this without using any enzyme. And I'm going to show you that if you put this just into water droplets, believe me, if you put pyruvate into bulk water, nothing happens. People know that. But if you make little water droplets and spray it into your mass spectrometer, you see lactate. And I'm showing you this here very clearly. You see the change. We've seen this in a number of cases. And now I'm showing you as a function of distance, different traveling distances. And you can actually do a calculation of the redox efficiency for this process. We've gone through this. And I don't find math and electric any great fun. You have to study it yourself in general, but we can get the reduction efficiency. And I'm just going to show you, it goes up to like 90%. We really, this is the kinetics of 10 micromolar pyruvate in microdroplets and its concentration dependence. As long as the concentration is not too high, it really gets all converted from pyruvate to lactate. Now, okay, great. Let's see this happen in another circumstance, if I might. I hope I'm doing okay with time. Okay, well, let's look at I don't know how many of you have ever made gold films or silver films. I'm going to talk about a gold film. You start with chloroauric acid. Here's AuCl4 minus, and it's H plus. That's okay. And you have to have some reducing agent. The first person who did this and knew what they were doing was Michael Faraday, who used lemon juice, citric acid, and indeed made colloidal gold. There were people who were making colloidal gold before and using it to stain glass windows and color them, but they didn't know what was happening. Michael Faraday understood the process. And you see, we have to change gold, which if you notice with this AuCl4 minus is in the +3 state of balance. It has to go all the way to Au0, okay, and form gold particles. Well, if reactions are being so accelerated in droplets, I said to my group, let's just do this. We'll just bring together 2 droplets, one that contains sodium borohydride, the other one that contains the chloroauric acid, all dissolved in water. We'll take a glass side and we'll look. And you see here, this is published in 2018, the production of the gold particles just using looking at it on a microscope slide. You do not need any mass spectrometer for this. You just watch it grow, okay? And if you look at how it compares in bulk solution, it's taking minutes, you see, for this to happen. Here it's happening in fractions of a millisecond. And the accelerated growth of the particles is by a factor of about 10 to the 5th. You've already seen this now, huge accelerations in droplets. And I said, before we publish this, we have to replace the sodium borohydride simply by water. And I said the effect would go away. And so here we are, we're doing this just with water. And to my surprise, they came to me, the people who did this, people like J.Q. Lee, Devlina Samantha, okay? And they showed me, look, here it is. Look what we see. We see gold nanoparticles being formed. So we don't need the water at all. We just spray this alone, and we did. We sprayed it alone, and this is a reducing agent-free synthesis of gold nanoparticles and microdroplets. We see also nanowires, and we had a lot of trouble getting this published because people said, how can you get reduction? Where's the electron coming from? Okay, and indeed, we think it's coming primarily, if you think about it, it's not coming off of water, it's coming off of OH minus, we think. And the intrinsic electric field at the air-water interface may be responsible for the spontaneous nanostructure formation and why we can grow wires and such like this. And now collaborated with Professor Wei Min and his brilliant graduate student, Shang, who's going to go on to Peking University, actually. And we published just recently, look at the date, 2020 again, that indeed it's possible to see a very big stark shift, okay? This is their technique for measuring it, which I'm not going to take the time much to explain to you, except that it uses both IR and fluorescence to be able to see the effect, okay, in this process. This is the so-called SREF spectrum in bulk water. And we see the difference in a big shift. And from this, you can calculate the electric field. Okay, so if this is true, what I'm telling you, it has amazing implications. It suggests the possibility, okay, that you really have OH at the interface. If you're, after all, if you're removing an electron, you should have, from OH minus, you should have OH. OH is called the hydroxyl radical, and that's highly reactive. That's what, of course, in the troposphere, the lower atmosphere, is used to clean things up. Okay, and we want to now show the visual proof of reduction at the droplet periphery. So here is an oxidized form of this molecule. Here's the reduced form, and here is taking a picture of it with a confocal microscope. And indeed, we see that it is really right at the surface of the droplet. We see this happening very much. And now, if this is so, we should be able to trap the OH radicals. And we use salicylic acid to form these different dihydroxybenzoic acids. And again, our mass spectrometer could pick this up and show that this was happening. So what is happening, if you believe what I'm telling you? What seems to be happening is that water is falling apart at the interface to form H plus and OH minus. I've told you that, and you've seen evidence of that now, I hope. And that the OH minus can, with the action with the electric field and with something else, lose its electron, leading to OH, which we've seen. And if the OHs swim around in water, they will find each other, and the two radicals will recombine. And they will produce HOOH, which is called hydrogen peroxide. Normal water doesn't contain hydrogen peroxide, and I'm asserting to you that water droplets do contain hydrogen peroxide molecules. And let's now try to get some visual proof of this. So people at Berkeley were able to synthesize this, and then Walker in my group followed the same thing and made this compound. I'm just going to give it the name PF1. And you see with the boron, and if you clip the borons off, you get fluorescein, which is fluorescent. Okay, we recognize that. And that's what H2O2 will do to it. So here it is. We're going to put 10 micromolar PF1 in bulk water. And here is the fluorescence. I see nothing. Here is the bright field. Okay. Here is actually an air-water interface. I still see nothing. Okay. Doesn't happen in a flat surface. Now, what about droplets? And indeed, if I look at PF1 only in aqueous microdroplets, look what I see. And I illuminate them with some type of light that causes fluorescence, UV light. And look, they're there. And let's go further. And so there's hydrogen peroxide there. Let's make a study as a function of droplet size. And that's what this shows. I hope this can be seen to you well. There are 3 droplets here, A, B, and C. The first one, let's look at what they are. A is 160 microns. That's its diameter. It's big. Here is B, 50 microns. And here is C, which is 16 microns. And look, hardly any green, very slight amount of green I can make out. Just becoming here, very bright here. And it's just showing you a graph of it. So small droplets, micro droplets, really take off in terms of this production that I'm telling you about. And I think I can best show you this. And here we are with a publication in 2019. I want to show you a movie, if I might. I hope this will work. Let's make this go again. Come on. Here we go. And we have here a test strip. Okay, it's running. But is the movie playing? Here is the peroxide test strip. Here's the sprayer. And here, if you look carefully, there's a little wet here. Droplets are coming out. Now, this thing contains horseradish peroxidase and a dye. And if this thing had hydrogen peroxide, it would turn blue. Okay, and if you look carefully and be patient, this is played times 10, because otherwise it's like watching paint dry. Watch, it's turning blue. It really is showing you that you are producing hydrogen peroxide from this process. Okay, we have other tests to make it quantitative. You don't get much, but it's there. So there are all types of reactive oxygen species at the water droplet. Okay, let's go on now to the next slide. Let's actually talk about one of its uses. This is the application to organic synthesis. This is done by Dan Gao and her husband, Jin. And this is going to be that aqueous microdroplets containing only ketones or aldehydes undergo what are called Dakin and Baeyer-Villiger reactions. And I'm showing you the Baeyer-Villiger reaction here, in which an O atom gets inserted. And people normally use hydrogen peroxide to do this, or some other peroxide. Other peroxides tend not to be so stable. Hydrogen peroxide is better. And we're generating it automatically in the droplets. So we just need to spray them. And indeed, we've done that and again, published this. And this is the BV reactions in microdroplets. And you can see that it really does happen. Now, people said, well, it must be that it's all due to friction. Somehow you're charging up the droplet because you're making it move in your silicon tube, and that it's all silica tube, and that's what's causing it. And I'm going to show you it's not friction. It's intrinsic to the droplet. And so this is now published, again, just last year, 2020. And I have a movie to show you, if I might. If I were with you, I'd actually demonstrate this to you with a tea kettle and a glass that contains some ice in it. You would see the outside of the glass would get coated with droplets, and they would contain hydrogen peroxide. Let me show you this. Let me play the movie. Here we go, I hope. We're bringing up now a plate. This is a silicon wafer, okay? And we have just humidity in the air. And if you look carefully, because this is a cooler, it's condensing and we're forming droplets. And if you look real closely, I think you'll start to see that, you know, they're growing. It's 5 times faster now. And if this is so, we're not rubbing anything particularly, okay? And now let's take our test strip. You can see the droplets pretty clearly, I think. Here we go. Here's the hydrogen peroxide test strip. In water, it does nothing. Now we'll take it over the surface. Okay. And look, it contains hydrogen peroxide. And we've been able to also show that Dr. Maria Dulé is particularly the killing of bacteria with these water droplets. It really is a bactericide. So I've done that. And so I now want to close this lecture, and I ask you to indulge me while I close this lecture with some wild speculation. What I tell you next is a work in progress, but I think you'll see that it has an amazing connection to what's going on about us in the world today. This is really trying to answer the question, can we learn anything from a water microdroplet? Can it teach us anything? And so here's a review article that came out, and please notice the date, 10th of February, 2021. We're trying to make this talk really, this is the first time I'm presenting this in a talk, as you can imagine. And this is a review written by these people who are actually in Slovenia, okay? And this is called Relative Humidity in Droplet and Airborne Transmission of Disease. And I want to show you about this. You may know this already, but they have looked at all types of respiratory viral diseases, influenza, adenovirus, something called RSV, and the coronavirus. And there's the ones in blue are the winter. This is the summer. This is the data from Paris, France, but it's true all over the world. That you see it peaks in the wintertime and it falls off in the summertime in terms of the disease. Of all these viral diseases are that way. Okay, that's a fact. Now, whether this is just a correlation or whether there's some causality, well, the people are arguing about that. They don't know for sure. And I want to show you some more things that are going on. How can outside temperature affect what's happening? And the answer is actually indirect. And the indirect is that when it is cold outside, we keep ourselves warm, at least very much so in where I live, by indoor heating. We heat the air one way or another. And the indoor relative humidity changes. The relative humidity, and this is showing you the months, January, February, March, April, May, June, etc. And it's showing you, this is data from Gothenburg, Sweden, that it goes to 50% in the summertime and it drops down, okay? We see the difference between 30% and 50%. And this is the outdoor temperature. And the temperature here is going, and this is centigrade, from around 20 to towards freezing, okay, 0. And you see how it's changing the humidity of what goes on inside. And so there seems to be a strong correlation of relative humidity. And we're finding this in the production of reactive oxygen species in water droplets. That's work that's not yet published. And so now I want to share with you a research hypothesis, if I might. Previous work does not emphasize what is the chemical contents of exhaled droplets when you yourself breathe out droplets, or what droplets are like. It is hypothesized the droplets contain compounds thought to be reactive oxygen species that kill bacteria and viruses. I really have evidence of that. So recent work in our lab have definitely shown the existence of such species and even their ability to punch holes in the cell walls of bacteria. And we know for sure that H2O2 is present, and in a saline environment, a little bit of salt in the water, we suspect that you're going to form HOCl and OCl minus. Some of you might recognize that's what you call bleach. That's the hypochlorite anion. It's also present, okay? And if these reactive oxygen species, such as H2O2 and HOCl, rapidly inactivate viruses in droplets of 20 microns and smaller, then the fact that these droplets remain airborne doesn't matter very much as to what their size is. What it's telling you is that they will happen rapidly, and that if you can make small droplets, and they work best at this higher humidity, as I'm mentioning to you, to make this effect, then indeed you're going to kill your viruses. However, the rate at which droplets reach their equilibrium state matters, as the chemical reactions need time for the burst of oxidation agents to do their work. And so relative humidity does not matter for end state size. It may be critical to determine the viability of the virus inside the droplet when the droplet reaches its end state. So what I'm now leading to is a wild thing to mention to you. You often hear people tell you with COVID-19 that you should wear a mask. And they say often to you, well, the mask, you see, protects you from particles that come from the outside into you. And I'm telling you that the mask actually helps you in terms of making the humidity the right levels and the rest, so that you kill stuff that's in your mask. It also has another effect, but no one's talking about that much. And so I think now you have a sense from this, I've only touched on a couple of things, why it is that I love microdroplets. And if you want more information, here is my email address. You can write me, and I'd be glad to take any questions that might come up. And so I think I'm going to stop sharing the screen here, and very much appreciate this opportunity to tell you about this type of work.
Thank you, Professor Zayar, for such a fascinating lecture. So would you like to take some questions?
Very much. I'd be happy, or comments or whatever people say. You notice this doesn't simply fit into any one category very well. It's not clear what it's not clear what form of chemistry this all is. Okay, it has medical aspects to it and other things.
Well, this is a very, very fundamental chemistry. And what appears that now the microdroplets could become the most effective, reagents to inoculate places. Actually, one can use these to, instead of spraying high concentrations of hypochlorite bleach or other kinds of reagents, if this can supply the dose, this could be antibacterial and antiviral, perhaps.
We're very interested in exploring that.
Mm-hmm.
And I'm enthusiastic about that possibility. And okay, here's a question that's written, something fascinating. How does water get droplet nature inside cell? Well, there are in cells lots of droplets of various sorts. But I think you're seeing that the chemistry of the interface is very different than the chemistry in bulk water. I'm telling you that people are making a mistake. I'm being assertive here, and I'm telling you they keep using experiments done in bulk water and they're thinking that they apply to what goes on in the cell, and that's a mistake. And I think, as I'm trying to show you, very different environments. Interfaces really matter. This is interfacial chemistry.
Yes, of course. And in the air
Is there a
water
critical size below which the reactivity is no longer enhanced? I don't know that. I know it's, as I've made the droplet get smaller, it's gone up in terms of the effectiveness. Now, I don't know what happens below 1 micron much.
Okay.
Remember, 1 micron still has, millions of water molecules in it. This is not just a cluster. It's not a water cluster. That's not what I'm talking about.
What happens if you have little bit of oil droplet also there, because you're talking about air-water interface?
So we have done a lot of water droplets in oil, and we see these effects again of the droplet being so charged, and that you can make chemical reactions go much faster. This really interests me. Another part of this interests me as to whether or not this can be made into some type of form of preparative chemistry. I'm very interested in that possibility. And Nanamani, who's at IIT Roorkee, was indeed trying to work that out with us, and might be continuing that.
Any possibility of interfacial catalysis?
Yes, indeed. I was showing you can make all types of metal nanoparticles, and I believe they can also be used for catalysts. And you're getting these acceleration rates. You can think of itself as water being a catalyst, which is normally no one says, but the way it's acting is that way.
Is there any question on that? Yeah, there are many questions in the chat window. If you you can see them.
So here's one that says, the electric field generated by the droplets are very high. So is the Stark effect suitable to describe this as nonlinear e to the nth effects will also be important? Interesting question. I'm at the moment, I've been interpreting it simply as a Stark effect, but you have to calibrate it, and I've been acting it that way. I don't know fully the answer to that question. It is possible to correlate the size and reduction efficiency of microdroplets. And can this be used for some selective functional groups? I'm very interested in this possibility. Look, no one owns droplets, microdroplets, and there's so much to do here. Please, if you're interested, try it. Try to do things. We're finding all types of new things every time we do something. There have been surprises in this. So I urge you, just get involved. There's no way that any one group can do all the things that are possible here. And no one owns it, okay? Great. I'm not good at taking all these questions, maybe somebody else. Okay. Let's see. So it's very easy, as you see, to spray these droplets. And I now have an idea that fog and mist is actually good. That some people who take steam baths, it really actually is very healthy. And I think I'm beginning to understand partially why. And I you know, people had told me this and I never paid much attention, but now I'm beginning to think there's some real scientific basis to this, a chemical basis. I don't know. More to be done. Hi.
Hi, Dick. A quick question. Just thinking about the reaction you're proposing where you are generating the hydroxyl radical and H2O2 that you see in your dye. If I do a mental balance of the charge, I don't know what's happening to the protons. Where are the protons going? Because you're generating free protons in the process of oxidizing water to H2O2.
I don't know where all the charge has gone, but the thing starts as neutral and stays neutral all together. But there's a separation of charge, and there are charge gradients that are in the system.
No, so when you come from H2O to H2O2, you need to absorb 2 protons. Is that your dye picking it up, or in the case of nanoparticle, the reducing agent that's picking it up, or?
I don't know where the electron altogether goes. I'm wondering if it isn't solvated electrons is one possibility. That's the question: is what happens to the electron?
I was thinking you were going to say that because that's what it was all
Yeah,
about.
I don't, or it's gone on to the some impurity like the bicarbonate or something else that's being changed. I don't fully understand what all I'm seeing, and I want to admit that to you right away. So,
It would be lovely if it were solvated electrons in a microdroplet. That would have been something.
and incidentally, I think you can start to appreciate why trying to get any of this work published has been very painful, because I get back reviews that tell me how exciting it is and then how unbelievable it is.
Isn't that you?
Hello, I have a question.
I think
Please.
Yeah. At somewhere you are presenting that the digestion of protein with the water droplet, and then you show the correlation with the diameter, and then you analyze by mass spectrometer.
Yeah.
It was showing that, you know, as soon as you are going smaller and smaller droplet, the digestion is increasing in terms of mass spectrometric data. But sometimes I find that correlation is not quite linear. Sometimes it is increasing, sometimes it is decreasing. Can you make some, sometimes the correlation, as the diameter of the droplet decreasing, sometimes the correlation between the, you know, the number of digestion of the protein in terms of mass spectrometric data is not quite, you know, inversely proportional. It is sometimes it is increasing, sometimes it is decreasing.
I've not seen it. I don't know about the decrease. I've always seen that smaller droplets are more
As for example, for 2
But
nanometer,
it's not linear.
not linear.
Not linear. That part I know. It's not linear. But it is only in one direction. The smaller the droplet, the more effective.
Yeah, yeah.
Is what I've seen.
Yeah, and the second thing is that I'm really got fascinated when you show the solution of the Gauss equation to present the radial distribution of the charge. The radial distribution of the charge solving the Gauss equation. Basically, from the electrodynamics, we know when you put some charge, the always charge come on the surface. If you put some charge, particularly electron, the electron always come to the surface.
Right. If you have an unbalanced charge, they repel each other and they always go to the surface.
Surface, yeah. So so that's what also you show in your graph for the solution of the Gauss equation.
But I want to again stress to you that this is because there is some chemical interaction between the positive charges and negative charges that is different at the air water or oil water interface.
Yeah. So you have final conclusion at some point, 3 points. So it is dissociating H plus OH, and then OH is releasing the electron, and that electron is coming to the surface, and so that's why your solution for the Gauss equation is closely matching with your experiment, isn't it? Should I repeat my question? So you have 3-point conclusion. One is water droplet dissociate as H plus OH minus, and then OH release the electron, and that electron come on the surface, and it is essentially moving around. And the same thing you are trying to solve from the Gauss equation, and Gauss equation always tell that if you have excess charge, it come to the surface. So
right.
So the correlation is quite, quite known probably 200 years back, but now you are really demonstrating with the chemical examples. Is it like that? Will you say this way, or you think?
Certainly, in Gauss's law has been understood for a long time. I agree to that, but I'd not seen before the application this way
For the chemical system.
Yeah.
Yes. Yes.
And I haven't talked about it, but it turns out because you have such a strong electric field, you actually are setting up some form of electrochemical cell in your droplet.
Yeah.
You're actually doing electrochemistry of some sort.
And
I'm seeing these redox reactions.
And one problem.
I don't have an electrode where I'm applying a potential.
Yeah. One problem for the solution of Gauss equation is that it is close to Poisson equation, and Poisson equation do not have, boundary condition is not always known. And because of that, sometimes solution become unphysical. So I think you are taken care while solving that. The Gauss equation has a problem. Its boundary condition is not really known, and sometimes it gives unphysical solution when you solve it with a wrong boundary condition. I'm sure you have taken care about it. That's what my point. This is a comment, really.
Thank you.
Yeah. So it is essentially Poisson equation. Poisson equation has a problem. It's boundary. If you choose wrong boundary condition, it gives wrong solution. But I'm sure in your case, you have chosen proper boundary condition, and that's why it presents the correct solution which mimic your experimental result.
I really find it close mimicking, close behavior. Yes.
Yeah. So
Any other question?
thank you.
2 more questions. Okay.
2 more questions from the chat window, please.
Okay. Can you read them, or I can? What do we have?
So enhancement should be observed even to microdroplet from other polar liquid. That is obvious.
Let's see.
think one is that
Some selective functional groups, we've talked about that. Yes, Professor Sasson Sheikh in Jerusalem has a model to catalyze reactions by external electric fields. So can we have designer reactions catalyzed inside droplets? And my answer to that, this is Professor Datta is asking this question to everyone. And my answer to that is absolutely yes. And I didn't give you all that chemistry, but there really are examples of that that are happening. And I think that the microdroplet is allowing us to actually apply an external electric field to drive chemical reactions. You know, my colleague, Professor Boxer, has very much made a career out of showing that catalysts work by the electric fields that they make in their cavities. And I'm telling you, the droplets are acting this way too.
Professor Jare, there is a question from Manik Pradhan. Question is from Manik Pradhan. You can see. Manik Pradhan.
Yeah, I'm looking to see if I can find him in this. Let's see what he writes. Here he is. As H2O2 occurs in ambient air, is there any link with human exhaled breath? Manik, yes, we have seen in exhaled breath, there's hydrogen peroxide. That's what I'm telling you.
Okay. Thank you.
Much. And you can, and it's only parts per million. It's not large, but it's there.
Okay.
And this is interesting. Good to see you.
Nice to see you. Thank you.
Thank you, Professor Zayar, for such a fascinating lecture. And I think certainly this one of the most fundamental finding that you have described. I think many of us have enjoyed it thoroughly, and we look forward to having much greater success with your endeavors. Thank you again. I shall be in touch.
I look forward to returning to Calcutta and enjoying your wonderful fish dinners that are possible and so forth.
Absolutely.
I miss you all.
Yeah,
thank you so much.
With your mist, actually, you can kill the corona, and I think all the pandemic would be over. And you.
Well, we're gonna try.
Yeah.
If I might comment to you about something that you perhaps haven't thought much about, is airplanes.
Yes.
Right now, airplanes are priding themselves on how many times they turn the air over in the airplane. And if you've taken a long trip in an airplane, which I have when I fly from the US to India, you find you dry out. The relative humidity is being held very low. And I'm also trying to explain to you that is absolutely the wrong thing to do to stop disease from being communicated between people. You want to increase the humidity, not decrease it.
Okay, that's a very important observation.
Okay.
I think I will be in touch with you through email for certain formalities. And
well, I appreciate it again. So thank you so much for having me.
And again, we are profoundly thankful for delivering lecture. Thank you very much.
Bye-bye.
Bye.
Bye, Dick. It was nice to listen to you as always. This is
Thank you. Good to see you.
Hello, this is Sottomurthy. This is Professor Sottomurthy.
Yeah.
Yes, I see you. Oh, my goodness.
Yeah. So I was, it's wonderful listening to you, and I just sent you a mail. Yeah.
Thank you. And you're looking good. Great.
Well, thank you. And you are always energetic and enthusiastic. Yeah. Wonderful. Yeah. Thank you. Yeah, Satyajit alerted me just 5 minutes before your talk started, and I'm glad I could listen to you. Yeah.
Big surprise. Appreciate it. Great. Thank you.
It's my pleasure. Yeah. We look forward to seeing you in India soon, whenever that is.
I hope so. Thank you.
Bye-bye.
Bye-bye.
Yeah, bye, Dick. We can meet sometime in India.
Good to see you, Sati.
Yeah, it's great to see you. and I'm glad that Satyajit alerted me on this talk.
Yeah.
It's always wonderful to listen to Dick. Yeah.
I
Okay.
think we must meet physically as soon as possible.
Sure. Whenever it is, wherever it is. Yeah.
Yes, yes.
Take care.
Thanks a lot.
Yeah. Thank you, Satyajit, once again.
Oh, thank you. Thank you. Thank you. I think Saurabh also came in. Saurabh also there.
Kim?
Hmm.